Columbia University Lamont Doherty Earth Observatory in Palisades. He is a biological oceanographer who studies the growth and physiology of planktonic organisms in the relation to water quality issues such as nutrient pollution, sewage pollution, harmful algae blooms, and hypoxia in rivers, estuaries, and the coastal ocean. He has collaborated with Riverkeeper on water quality issues in the Hudson since 2006. He also conducts research in the Gulf of Mexico and the Arctic Ocean, and he lives in South Nyack. But it seems like you get around, Andy. So with that, looking forward to your presentation. Thanks very much.
Great. Well, thank you very much for inviting me. I'm going to share my screen. Let's see. Oh, well, give me a moment here. Okay. So now you guys should. How does that look? Oh, center this. You see the slide? Everything looks good?
Yeah, that looks good.
Okay, thank you.
Yes.
All right, so this out of the way so that I can read. Okay, so yeah, I've been involved in water quality sampling in the Hudson for about 15 years, and so I'm going to tell you about some of the history of how I got involved in this, and also some of the insights that we've gleaned from the samples that we've collected over a long time. So who am I? Well, I'm, as you just heard, a biological oceanographer, which is to say that I study life in the oceans, and that includes estuaries. And I'm basically a microbiologist in that I study very small organisms in the ocean, mostly single-celled organisms. And mostly these organisms, the things that I work on are organisms that are found in the plankton, as opposed to, let's say, things that live in the bottom. And I do this work as a Lamont research professor at Lamont Doherty Earth Observatory. That's a research campus of Columbia that's in Palisades, New York. For those of you who drive on 9W, it's on 9W just north of the state line, right before you cross into New Jersey. And there's probably something like 500 to 600 people working at Lamont, including around 200 or so PhD level research scientists. It's actually one of the larger employers in Rockland County, but most people are mostly unaware that we're there unless they've been to the campus for our annual open house that happens every October. So second week of October, you can mark your calendars. That's always on a Saturday. And it's always a lot of fun, especially for people that have kids. We do a lot of kid-friendly, science-y related stuff, and I encourage you all to visit. Okay, so I study microbial plankton from estuaries to the high seas and from the Arctic Ocean to the tropics, most of which is not relevant for what I'm going to talk about today, because since I arrived here I first came to this area in 2004, and pretty much since then I've been studying the microbiology of the Hudson River, which includes a lot of inputs of bacteria that don't normally belong there, and many of those come in through sewage contamination. And in a moment, you'll understand why I've done this work in collaboration with Riverkeeper, which is a local advocacy group that probably most of you already know. Okay, so brief outline of the talk. I'm going to say a few words about the history of how and why we got this partnership for water quality sampling started. And then I'll tell you some of the things that we've learned, starting with the things that we learned at the beginning, basic spatial and temporal patterns that were obvious within a few years of sampling. And that'll lead into some of the relevant side projects that we've done. And if I have time, I'll get into some of the emergent properties that we see now that we have a much longer data set. And I'll be focusing to some extent on observations that are relevant to the waterfront communities of the Tappan Zee region, partially because of the audience today, but also partially because, this is where I live too. Okay, so I've been doing this for a while, so I'm going to reminisce here with some greatest hits, if you like. So first of all, this work was initially motivated by a guy who some of you probably know named John Lipscomb, who is the Riverkeeper boat captain. So John would travel up and down the river doing patrols, and he would hear some version of the question, how's the water? From people up and down the river. And he really didn't have a particularly good way to respond. But he recognized that what people were asking was essentially, if I contact the water, if my kids or my dog jumped in the water yesterday, should I be worried? In other words, am I going to get sick from contacting the water when I swim or kayak or whatever? And a contact problem like that, that's really a microbiological problem. So if you contact the water and there are microbial pathogens, pathogenic, disease-causing bacteria and viruses in the water, you might get sick in the next few days. Okay, there's lots of other potential contaminants. In the Hudson, we all know about PCBs and heavy metals and other potential contaminants in the water and the sediments. Those tend to have other exposure routes and they present more chronic health risks. So you get sick because you eat the fish for a long period of time, or you drink water that comes from the Hudson. If you're in one of those communities that gets their drinking water from the Hudson over a long time, those contaminants can build up in your body and that can cause disease. But that's not really a contact related health threat. Whereas the microbiological problem, that's really an acute health risk in the sense that you might get sick, soon after you contact the water. Okay, so that was the basic problem that people up and down the river were asking of John, and he didn't really have a way to answer that question. So he came to Lamont with this very simple question. And to be perfectly honest, we didn't believe him. We didn't believe that there was no information available. Okay, and let me put that in context. So I arrived here in 2004. For the prior 12 years, I'd been living in San Diego, Pensacola, Florida, and on Cape Cod. And all of those places had really robust, publicly available data sets on swimming water quality. And of course, that's because those are places where there is a very strong tourist economy. And it's really important that the water quality be good and that it be publicized that it's good. And as it turns out, John was correct. There really wasn't publicly available water quality data on microbiological water quality. So the New York City DEP for decades had been collecting data around New York City. But they were at the time, they were only making their data available in highly aggregated form in occasional reports. So like 3 years later, you would find out that 3 years ago, the water quality was okay on average for the summer for some region around New York City, right? It's not really what people wanted to know. And then, once you got north of the Bronx, there really wasn't any publicly available data to be found because you were outside of the New York City DEP jurisdiction. But also, there really wasn't very much data collection, even if it wasn't being made publicly available. It just wasn't there. Nobody was sampling, or there was very little sampling going on. And this is a legacy of having a system that historically had such poor water quality that there was no need to sample, right? Everybody knew that the water quality was always bad, and so everybody knew you shouldn't really go swimming. But what people wanted to know is, had it gotten better? And everybody figured probably, but there was no way to know for most of the river. Okay, so that was the problem. So how do you solve that problem? Well, it's really simple. You start sampling, right? So we started sampling in 2006, and we sampled around, primarily around New York City and the Tappan Zee region, because of logistical reasons. I mean, we had no support, right? This was mostly a volunteer effort. It was done by John Lipscomb, who's pictured here, and Greg O'Mullen. Greg was being supported by a postdoctoral fellowship from Columbia, and so he had some ability, well, both of them had some ability to do this work kind of pro bono. Based on the data collections from 2006 and 2007, we were able to get some support from the Wallace Foundation that gave us some salary support, and also outfitted the Riverkeeper vessel. And that allowed us to do sampling from New York City up to Waterford, New York, which is, north of Albany. Right, so we were able to do the entire tidal estuary once we had the vessel properly outfitted. And so starting in 2008, we began sampling the whole system about once a month at about 75 stations. And the sampling that we did in 2008 set the pattern for what we were going to do for the rest of the time. So we measured a lot of different things, but the primary health risk tool that we were using was the concentration of enterococci bacteria. Okay, so what are they? All right, so we're interested in quantifying the health risks associated with sewage and fecal matter that is in the open waterways. So how do we do that? Well, it's pretty straightforward. Conceptually, right, we all eat. The food enters our body, it goes through our digestive system, and along the way, it picks up some bacteria. And some of those bacteria may be pathogenic, particularly if you're sick, but not necessarily. Okay. And then when that leaves your body, right, it goes out into the environment, ideally goes through sewage treatment, and it is okay. There's a particular subset of the bacteria that are picked up in your guts that we can measure, and we call those fecal indicator bacteria. Okay. And in the past, people measured fecal contamination of waterways using a group of bacteria that are called the fecal coliforms, and more recently, only a subset of that group, the well-known E. coli, are thought to be a better approach than the fecal coliform group, which is more broad. And alternately, there's a genus of bacteria called enterococci, and these are also recommended for this purpose, particularly in brackish water. And just so that you know, I'm going to be showing some data later on, and that's all based on data, essentially counts of enterococcus bacteria. Okay, so what are the enterococci? These are bacteria that are normally found in the guts of warm-blooded animals. Enterococci themselves are not usually particularly serious pathogens, at least not in an environmental setting, sometimes in hospitals, but they are indicators of fecal contamination because they're found in the guts of warm-blooded animals, but generally not found in open waterways. Okay, so if you find something in a waterway that has enterococci, high counts of enterococci in it, it means there's something there that was fairly recently within the gut of a warm-blooded animal, and hence it's a fecal indicator. So they can come from humans, they can come from other mammals, they can come from birds. It doesn't actually matter that much necessarily in terms of the health risks, but it's a really different management issue, right? Managing human sewage is very different than, say, managing birds or deer, right? So, okay. So enterococci have been recommended by the US EPA as a fecal indicator to protect human health from contact recreation since 1986. They're recommended for use in fresh and brackish and marine water quality assessment. And so because they're good for fresh, brackish, and marine water, we decided to use enterococci for the whole system, right? Because we were sampling from the marine end in New York City up to the freshwater parts of the Hudson, and we wanted the same indicator throughout. The other thing that was going on was that, back in 2006 to 2008, when we were sort of figuring out how to do this, New York State was the only state that was still using fecal coliforms as a water quality criteria for swimming. And we figured, well, that's obviously going to change pretty soon since the EPA had been recommending that change since 1986. And we wanted to kind of get ahead of where the state was going to be in a few years. So we went with Entero for several reasons. As it turns out, it's taken a little longer than we might have expected for the state to make that change. But anyway, that's what we decided to do. Okay, so we started collecting samples in 2006, and then in 2008, we expanded to sample the entire tidal estuary. So this is a map showing all of the locations where we have routinely sampled around New York City and then up to north of Albany. Okay, 75 stations, more or less, approximately monthly between May and November. We measure enterococcus, and then we measure a bunch of other things like temperature and salinity and oxygen, and turbidity, chlorophyll, whatever, the usual things. And most of the sampling has been done from the Riverkeeper survey vessel, the Fletcher. Okay, and then the other thing that happened was starting around 2011, Riverkeeper began involving citizen scientists in watershed sampling. So, we're on that vessel, and so we are restricted to the deeper parts. Well, we're restricted to the estuary. We can't go up above the tide in any of these watersheds. But people wanted to know like what's going on in my particular part of the system. And so that's where Riverkeeper started involving citizen scientists. They would collect the samples and then they'd started by bringing them to us, and we would process the samples, and then eventually other places started processing those samples as well. And so within a few years, we'd reached a point where this really was pretty unprecedented spatial and temporal coverage of microbial water quality indicators. Over 400 locations sampled at least 6 times a year, some of them much more than that. And critically, all of those data were being posted to the web within days. This website here is where all the data are available. The data pages here at this website get over 100,000 unique hits per year, which is one indication that we achieved our goal of making water quality data available to the public. And it's also a sign of how much people care about water quality. Okay, I have to say a few things about how you interpret an enterococci data. This is based on US EPA guidelines. So, there's different ways you can look at data. So if you're interested in, like, let's say the average or the typical values that you get at a particular location over time, the EPA recommends calculating what's called the geometric mean, the temporal geometric mean, which is kind of a weighted average of samples that are collected at the same location over time. And the guidance that they provide is that you should not have a count that exceeds about 30 to 35 colony forming units per 100 milliliters of water. I'll come back to that in a second. If you do any water quality sampling, you realize it's pretty variable. And so there's also a standard that's related to occasional spikes, right? And so they recommend that no more than 10% of samples should exceed 110 to 130 colony forming units per 100 milliliters of water. Now, both of these criteria are based on repeated sampling over time, and that's not that useful if you're trying to manage a beach. So they also have what's called a beach action value. So that's a number that you should close a beach to swimming if any single sample exceeds 60 to 70 colony forming units per 100 milliliters of water. Okay, so those are the 3 criteria that they have recommended. I have a couple of important comments on these criteria. So first of all, what is a colony forming unit? That's just an operational way of saying the number of active cells that are alive and that could grow under the right conditions, right? We're interested in things that might cause health risks, so they have to be alive. And then note that there's a range for each of these criteria. It's not a very big range, but there is a range. And the EPA provides discretion to the state or other regulatory agency to pick where they want to be within this range, basically. Okay, and then, let's consider this particular criteria, right? You're not supposed to have more than 10% of your samples exceeding, about 100 cells per 100 milliliters, right? So that's a high count. 100 cells per 100 milliliters is a pretty high count, right? That's something you should only exceed rarely. So 100 per 100 milliliters, that's the same as 1 per milliliter. And so within that context, if you were to count all the bacteria in a milliliter of water from the Hudson River, what you would find is that there's about 1 million to 10 million bacterial cells, right? And so when you have one enterococci out of every million other bacteria per sample, that's high, right? So we're talking about very, very rare organisms reaching a level where we would be concerned. And so because they are so rare in any given sample, we typically have to detect them by growing them up relative to other bacteria. And so that means that there is typically a delay of about 24 hours between collecting a sample and then finding out how many enterococci were there in that sample when it was collected. So day-to-day management, right, can only provide information on water quality yesterday, which is not exactly what you want. There's not much you can do about that at least with sort of the standard techniques. And that's why there are guidelines that are based on longer term values. That's why these guidelines exist. Okay. Another point is that all three of these criteria are based on the health risks associated with full immersion swimming, right? Getting into the water completely. Other kinds of contact have different risks, and the risk levels are different. So these criteria do not apply exactly, or maybe not even at all, to wading, boating, fishing, kayaking, sailing, and so on. All right, but let's be clear. I am not a regulator. It is not my authority. It is not my job. I'm telling you about these criteria to give you an idea of what is good water quality versus bad water quality. I'm not saying, oh, this is in violation of anything, right? It's just that these are guidelines. Someone else's job. Okay, and then last point is I'm going to present a lot of data as either in the form of geometric means and also this statistical threshold value. And so to calculate these metrics, the EPA recommends collecting multiple samples from any given location, at least 5, over 30 days, and then estimating, let's say, the temporal geometric mean or the statistical threshold value from repeated sampling at a given location. Okay, we don't collect our data like that. We can't collect our data like that. We're interested in sampling the whole system. We can't do it 5 times a month. So what we have done instead is we have collected data from the same locations over many years. And from that, we can get a very good understanding of what the typical distribution of values is for any given site over time. And from that, we can estimate a geometric mean or a statistical threshold value. It's not exactly what the EPA says you're supposed to do. I would argue that it is statistically equivalent, but it doesn't matter because, remember, I'm not a regulator, I'm not a manager, it's not my job. I just want to give you an idea of what is good and what is bad, okay? All right, so let's get into some data. That's all preamble. The interesting part is about to come up. Okay, so the very big picture, right? The solution was to begin collecting those data, make them available within a couple of years after we started a number of really obvious and important observations were easy to make. And so I'm going to present a series of figures that look kind of like this one. There'll be some sort of a graphic that's based on the beach advisory value. So these are percentages of samples, of total samples that have been collected. The ones that are exceed the beach advisory value are red, and the ones that are acceptable are green. There's also the geometric mean and the statistical threshold value, which are also coded green or red, depending on whether or not they exceed the threshold that is set by the EPA. Okay, so if we take all of our data together, collected over all years for the entire Hudson, which is kind of a little excessive, but nevertheless, really important. What we see is about a quarter of the samples that we've collected over time had high enough enterococcus counts to close a beach for swimming. Okay. Conversely, what that means is that about 3/4 of the samples were okay for swimming. And that's probably better than most people think. It's still probably unacceptable too often, right? That's a frequency equivalent to about 1.5 days per week. And I think most people would say that that's too frequent. But anyway, it's probably better than a lot of people think. So looking at this in a little bit more detail, that's, you get a certain picture from looking at the beach advisory frequency. If you look at the overall geometric mean, it kind of agrees with the breakdown of, well, it definitely agrees with the breakdown of samples, right? Most of the samples are okay. And the geometric mean standard, sort of the typical conditions you would expect to find on average, right? Also okay. It's 16. It can be as high as 30 or 35. However, the statistical threshold value is exceeded 16% of the time across the entire system. So what that tells us is that actually typically the system is fine, but there are frequent high values, right? All of these data are saying the same thing. Okay, I'm going to break this down into much more nuanced patterns, but like this honestly is probably the most important take home. And the other thing that I want you to take home is I want you to remember that prior to us doing this work, there was no basis for making any statements like this for most of the river. And certainly no basis for making statements like this using data that was publicly available. Okay, the other thing that we noticed, and this is not a shocker, is that the preceding weather had a big impact on the counts that we would see. And this is true everywhere in the system. So again, this top figure here is summarizing the entire system, all weather put together, but we can also divide our samples into samples that were collected during or following what we would call wet weather or following what we call dry weather. What do we call wet versus dry? Wet is if it has rained at least 1/4 of an inch in the 3 days prior to the sample being collected. That's wet. So today would definitely be wet, even though where I am, the sun is shining. It rained previously, so that's a wet weather sample, and dry, I think, is obvious. Okay, and there's a much higher frequency of exceedance for the beach action value. The geometric mean goes up, and you have more frequent exceedances of this high level. Okay, and it's the opposite during dry weather. Okay, and that's because rain increases release of sewage, right? That's mostly because of overflows or bypasses of the sewage treatment plants, or and stormwater runoff from everywhere. There are also regional patterns. I don't know why this graphic is so, sorry, this graphic is very hazy for some reason, but I think you can still see it. So you can divide the system up into different regions, right? So Capital District, this is what we call the Catskills region, Mid-Hudson around Poughkeepsie, the Hudson Highlands region, where we are, we're calling that the Lower Hudson or the Tappan Zee region, and then around New York City. Okay, and same kind of figures, right? And sure, the Capital District actually is different from the other districts. It's the one that is most frequently unacceptable relative to other districts. And the Mid-Hudson region, right around Poughkeepsie, is the best in terms of frequency of exceedances of the beach action value, as well as the geometric mean and the statistical threshold value as well. So this is the one that is the best. This is the one that is the worst. But to be perfectly honest, there's a lot less variability than we expected, right? So there's a lot of people in New York City. There's still a lot of people in the Tappan Zee region, but a lot less. There's not that many by comparison in the Hudson Highlands region, and certainly in the Catskills region. And yet, these 4 regions are pretty similar to each other. That was surprising. We also sampled different kinds of locations. So we sample in the middle of the channel in a number of locations. And we also sample near shore. And there's not that much difference between what's going on in the middle of the channel and what's going on near shore. But there are other kinds of nearshore locations. So we sample at a number of locations that are near a sewage treatment plant outfall. And those, perhaps not surprisingly, tend to have a little bit worse water quality from a microbial perspective than other nearshore regions, other nearshore stations. And we also sample in the mouths of a variety or of a number of tributaries. So, you have tributaries coming into the Hudson. Where the tributary enters the Hudson and there's tidal mixing, that's what we call a tributary mouth. And actually, all of those tributary mouths, all of the ones that we sampled, had more frequent high entero counts than basically any other kind of sample type, including near sewage treatment plant outfalls. That was a bit of a surprise. And so we asked the question, are tributaries to the Hudson more frequently contaminated than the Hudson itself? And answer is yeah. This is where the volunteers came in, because we couldn't sample, we're working from this vessel, we can't sample above the tide, but lots of volunteers went up into their watersheds and collected samples and then brought them back to us for analysis. And what we found was that in the non-tidal regions of every tributary that we sampled at, the counts of Enterococcus were considerably higher and much more frequently above this beach action value, as well as the other metrics, than the rest of the Hudson, than the main body of the Hudson. And so, so every tributary that we've sampled, is more frequently contaminated than the Hudson into which it flows. I can break that down for you a little bit into individual tributaries. Starting from the north, you have the upper Hudson River, right? That's the part of the Hudson that's above the tide, north of Albany, north of Troy, really. The Mohawk River, which is the biggest tributary to the Hudson, Catskill Creek, the Roloff Jansen Creek, Esopus Creek, Rondout Creek, Wallkill River, the Mechanical River, which is in Westchester County, Sparkhill Creek, which is in Rockland County, the Sawmill River, which runs through Westchester County and enters the Hudson at the Yonkers waterfront, and then the Bronx River that enters into the East River. So these are all tributaries, and they're all, the red parts of these bars are much bigger than we see almost anywhere in the Hudson, including like places like Newtown Creek and Gowanus Canal. It's true in terms of the beach action values. It's also true in terms of the geometric means. These are the geometric means for each of these watersheds. Well, not really, each of these water bodies, right? And again, we're accumulating data, along the entire length of each of these systems, wherever we've been able to get samples from. And the geometric means are all pretty high. This little red line here, which I hope you can see, is that EPA threshold for the geometric mean. And only one of them is below that. They're all above it by quite a bit. And unlike in the Hudson, there does seem to be some kind of a relationship with human population density, right? In that the tributaries that are kind of more north, where there are fewer people, are lower, still contaminated pretty frequently, but lower than further south, where there are more people. Okay, I was not involved in the collection of these data, although I processed some of these samples. These data were collected by many, many volunteers, and they really deserve a great deal of gratitude. It's a sign of how much people care about water quality, particularly, obviously, where they live. Okay, let's take a look at where we live. This is the Tappan Zee region. These different colored dots, ignore the colors on these dots. These are just the locations where we actually collect samples within the Tappan Zee region. They are named here, and we have these red bars and green bars. And you may want to focus on any individual one of these because you live near it, but let me just say before you do that, let's just take a look at how variable this is, right? So all these different locations are pretty close to each other, but they're quite different. So some locations are as low as 3% of the samples are unacceptable relative to the beach action value. And then the highest one is here, 55%, more than half the samples that we collected. The regional average is 18%. Some of this variability is explainable, right? So all of the mid-channel locations that we sample, right? Like Stony Point mid-channel, Haverstraw mid-channel, Tappan Zee mid-channel, right? These are all very low. So if you are further from the shore in the middle of the channel, chances are pretty good that the water quality will be a lot better than close to the shore, in this region at least. And then we also sample at 2 different sewage treatment plants, right? The Yonkers sewage treatment plant, which is down here, that actually has a very low frequency of problems. That's different from the Orangetown sewage treatment plant, which is right there at near the end of Piermont Pier is where that outfall is. That one is much more frequently contaminated. So sewage treatment plants are quite different from each other. And then another thing to note, like this particular location here, the Sawmill River, right? That's right here in the water at the Yonkers waterfront. That's where the Sawmill River enters into the Hudson River. And that is a very frequently contaminated location, but it is right next door to 3 other locations, right? Irvington Beach is just a little ways up the river. The Yonkers sewage treatment plant outfall is right downriver from the Sawmill River, and then the Yonkers mid-channel station is just offshore of the Sawmill River. And yet these locations are pretty good, even though this one is really contaminated. And the same thing happens here at the Tarrytown Marina, right? There's the Tarrytown Marina, and the Tappan Zee mid-channel station is right there. Big difference between these 2 sites. Okay, that suggests that sewage contamination tends to be pretty localized. Okay, so let's get into that a little bit more. So let's look at 3 locations. These are 3 locations that are relevant to Rockland County. This is the Nyack waterfront. This is the end of Piermont Pier, and this is the mid-channel station in the Tappan Zee that we sample at. It's actually in Westchester County, but you know, that represents sort of the main water body here. Okay, so what I've done is I've plotted the Enterococcus count at the end of Piermont Pier and at the Nyack I've plotted the Enterococcus count at the Nyack waterfront and at the Tappan Zee mid-channel station as a function of the Enterococcus count at the end of Piermont Pier. Okay, so these 3 locations are within 5 kilometers of each other. And if they were correlated with each other, we would expect the data to fall along a line here, okay? So the blue dots are Nyack, the red dots are the Tappan Zee mid. They're plotted against the Piermont Enterococcus count, and there are no data here. All the data are here and here. There's basically no relationship between what's happening at Piermont and these other 2 stations. So it really does seem, and you know, I'm going to say I picked these locations for this particular talk, but we see this all over the place in the Hudson. Okay, it does not seem like stations that are near each other are correlated to each other. And so what that tells me is that contamination is very localized. And that is a really useful fact to know. It means that local problems probably have a local source and therefore also a local solution. So the good news about that is if a community has poor water quality along its waterfront, that community can make investments locally, and there will likely be improvements that will benefit them. And it also means that if a community has a local water quality problem along its waterfront, it can't point to other communities and say, oh, it's those guys over there. It's those guys over there. It's the people upriver or downriver or whatever. It's not. It's a local problem. And that's really, really useful. There's more to it than that. So let's look at some other data that we've collected from the same data set, right? So before I was showing you the Enterococcus counts at Piermont, Nyack, and Tappan Zee mid-channel. And now I'm showing you data on temperature and salinity collected at those same locations. And when you look at temperature and salinity, which is what we use to track water movement, we see that actually there's a super high correlation between these three nearby locations. And that's not at all surprising because we know that the system has tides and currents, and that creates vigorous horizontal mixing of water. So the water moves around between these locations and homogenizes the temperature and salinity, but it's not apparently homogenizing the distribution of fecal indicator bacteria. What that means is that the bacteria, at least the fecal indicator bacteria, are not traveling with the water. How can it be that they are not traveling with the water? They can't swim. So answering this question led to a series of side projects, and that actually taught us a lot about the ecology of Enterococci after they enter a natural waterway like the Hudson. Okay, so we call this the extraenteric ecology of Enterococci, which is a lot of alliteration. But let me just summarize a couple of key points. So this took a long time to do all this work, but basically what we found is that Enterococci die super fast if exposed to sunlight. So like on timescales of a couple hours, you go from high contamination to almost nothing in a sample that is exposed to sunlight. So they die really fast if they're at the surface and it's a sunny day. Okay, but it turns out that most of the Enterococci that are entering into the Hudson River are attached to particles, something like 40 to 70%. Okay, so they don't stay at the surface. Particle-attached Enterococci and other pathogens not only die more slowly for any given stress, because particles are a favorable environment for them, but they also sink. So free-living bacteria don't sink, but particle-attached cells can sink. So the scenario is you have an input of something. I mean, it doesn't have to be a pipe like this, but you can think of it as a pipe with a bunch of bacteria coming in. The ones that are not particle associated, they remain at the surface. And if it's sunny, they will die really rapidly. But a lot of them are associated with particles. And those particles sink. And the Hudson is really turbid. So it doesn't take much sinking before you get to a water depth where the sunlight is no longer causing very high mortality. And eventually, because the Hudson is not really that deep, they hit the bottom. Okay, so if that scenario was true, if the scenario that Enterococci sink to the bottom, if that is true, then there's a couple of predictions that one could make. So first of all, we would expect to see accumulations of Enterococci in the bottom sediments. We would also expect to see higher sediment concentrations in places where the water is more frequently contaminated. So higher levels of water contamination should give you higher levels of sediment contamination, right? And then we would also expect to see higher sediment concentrations of Enterococci close to shore because it's shallow, so it doesn't take as long to sink to the bottom. And also because that's where most of the sources are. Okay, so these are testable. As it turns out, we sampled a bunch of locations around the estuary. Location names are given here, ranging from places around New York City up through the Tappan Zee region. And we found a lot of Enterococci in the sediments. It's really variable, just like in the water. but very high concentrations. So yes, there are accumulations of enterococci in the bottom sediments. And what we've also learned is that there are other bacteria that are of concern in the bottom sediments as well. So they continue to be an indicator of potential health risks. Okay, so check on this first one. Is there a relationship between the contamination of the water column and what you see in the bottom? In other words, are the cells from directly above going into the sediment? Seems like it. This is a plot showing the geometric mean concentration of enterococci in the water versus the sediments lying underneath. And I think you can see there's a pretty strong relationship there. So yes, we see higher sediment concentrations where the water above was more frequently contaminated. And then finally, there's a pretty strong decrease in sediment contamination levels as one goes into deeper water. So this is the geometric mean of enterococci and sediments that were collected under 20 centimeters of water, under 2 to 3 meters of water, and under more than 5 meters of water. And there's a strong relationship there. So yes, as you go further from the shore into deeper water, fewer enterococci in the sediments. So check, check, check. This seems like a pretty solid scenario for what's happening and explains, I think to a large extent, why you have primarily localized water quality problems in the Hudson. By the way, these reservoirs of sediment, enterococci, and other bacteria are very easy to resuspend, and they can recontaminate the water. So, like the action of, kids wading through the water is enough to resuspend the sediments, and that will also resuspend the bacteria that are in the sediments. Okay, so, the ecology of enterococci is actually pretty complicated, right? Particles come in, then they get attached to particles, and then they sink, and then they end up in the sediments, and the sediments can be resuspended. There's a bunch of different sources, there are a bunch of different reservoirs. One could therefore question whether enterococci are actually a good indicator of health risks in the Hudson. So are they? Well, we don't know. To be honest, the only way to know would be to conduct epidemiological studies, and that work has not been done. So the best that we can do is we can look at correlations between enterococci and potential pathogens, assuming that if they are correlated with potential pathogens, then they are indicating health risk, right? So, so we have done that, and I'm going to just go through this quickly. So first of all, we showed that that the counts of of enterococci are highly correlated with counts of antibiotic-resistant bacteria. There's two plots here. So we have enterococci in the water versus this is ampicillin-resistant bacteria on this axis, right? There's a pretty strong relationship there. And enterococci versus tetracycline-resistant bacteria there. So basically, if you have high concentrations of enterococci in the water, you also have high concentrations of bacteria that can cause antibiotic-resistant infections, like if you have a cut in your skin or something like that, or if you inhale an aerosol or drink some of the water accidentally. Similarly, I'm not going to show you the data, but counts of enterococci are pretty highly correlated with counts of other known pathogenic bacteria like Salmonella and Shigella and Vibrio. And also counts of enterococci are also pretty highly correlated throughout the system with counts of E. coli. E. coli is another FIB, as I mentioned at the beginning. It's primarily recommended for use in freshwater. But whether you're in freshwater or in brackish water, at least in this system, these two indicators are pretty reasonably correlated with each other. All right, looking at the time, let me try to bring this home with a quick summary. So even in the first couple of years of sampling, it was clearly apparent that FIB, fecal indicator bacteria, were found everywhere, highly variable. Some locations were frequently good, some locations were frequently bad, but most locations are just really variable. But not all of that variability was, random. For example, counts were much higher following rainfall at many locations. There were a lot of regional differences, but really those were kind of smaller than they thought they would be, although the Capital District is definitely the worst. In terms of locations, on a smaller scale, tributary mouths and some of the sewage treatment plant outfalls were more frequently poor than other locations. Oh, okay. And then over time, particularly because of the efforts of a lot of volunteers, we learned more about what was happening in other parts of the system. So for example, all of the tributaries that were sampled, and I haven't said anything about sampling in the East River, but all of the embayments around New York City and southern Westchester County have really widespread persistent FIB problems. And we also learned that there did not seem to be obvious teleconnections between stations within the Hudson. So problems are really much more localized than we thought they would be, which I think is actually good news. I do want to stipulate that that is true based on the data. That seems to be true for the Hudson. I don't think it applies in the tributaries. I think that there's much more transport in the tributaries. But in the Hudson, it seems to be much more localized than we would have thought. And we also learned a lot about the extraenteric ecology of fecal indicator bacteria. They're killed really fast by light, but they are associated with particles. And so both of those things play a really big role in their ecology. In particular, particle association causes sinking, and sinking to the bottom creates these reservoirs of FIB in the sediments that probably explain the localization of problems, but also create another problem because they can be resuspended. And right, we also learned that FIB do seem to indicate potential hazards because they're correlated with likely pathogens. Okay, I'm going to, looking at the time, I'm going to end it there. I could go on for hours. I'm not going to, but I'm happy to take any questions. Thank you.
All right, thanks very much. Boy, interesting to say the least, Andy, for sure. So does anybody have questions? Just all they have to just unmute and fire away.
I'll start. Am I
Am I
getting feedback here?
Hi, Scott.
I am.
You're-
Hi, Andy.
there's a little bit of, yeah, that's better.
Is that better? Thank
Yeah.
you, Andy. That was a great presentation. So my question's related to the tributary mouths themselves and the findings related to the tributaries being hot, relatively speaking, compared to other
Right.
locations in the Hudson. And so I'm wondering if you have any thoughts on source of fecal indicating bacteria in the tribs. In particular, I thought I recalled reading something. There was a eDNA microbial trackdown project in the Wallkill that found, I think, if I remember correctly, Wallkill was predominantly maybe avian, goose, I don't know. Rondout was people. So I guess I wanted to get your thoughts on that, and if you had the ability to do a next step on tributaries, what might that look like if you were to march up the streams a little bit to investigate further? So that's my question, or both my two questions. So thank you.
Yeah.
I hope they make sense.
Okay, those are great questions. You're right. There is quite a bit of variability in sources. It's not just that, one particular tributary may have a predominance of one type of source relative to another. That's true. But also within any given tributary, there are regions that seem to have different sources. I think this is consistent with the general theme that I hope one gets out of this, is that there's a lot. Most of these problems are fairly localized, and that means since you have widespread problems, the reason, if you have localized, if you don't have a lot of transport, and yet you have really widespread problems, that means there's a heck of a lot of sources. And that's really, oh man, that is the challenge, right? Because it's not like there's just a big pipe somewhere that you can turn off. Right. And in many cases, the source of the contamination isn't necessarily sewage. I mean, in the Hudson itself, I'm pretty confident that it's human. In many of the tributaries, there's always human, but sometimes avian is more important. And so it's a really different management issue there, right? What are you going to do about the birds? And that may not be.
I don't know, longer goose seasons maybe or something.
Yeah. Yeah, I don't know if it could be the geese. But yeah, it's very frustrating, I will say, for the people who have volunteered to collect the samples, because they know how difficult it is to solve the problem. Because it's like, instead of there being, one big knob that you can turn that will solve the problem, there's, 500 little tiny knobs. And if you just turn one of them, it's not going to have much of an effect. And so it's a very tedious process. I mean, you're more aware of this than I am, but yeah. I hope that.
It does. Thank you. I mean, we've been thinking about this,
Right.
and we can talk offline about this more, about what a next step might look like. And it's
Yeah,
still an active part of the conversation. And stay tuned. If we get some traction on that, we'll reach out. So thank you again. Appreciate it.
Yeah, great, Scott. Good questions. So Andy, I guess I had a couple too. I mean, how long will Intero live in sediment if there's no light?
It depends a little bit on the organic matter concentration of the sediment. So places that get a lot of sewage input tend to have really high organic sediments, and that's like the perfect environment for them to persist for a long time. And when I say long time, I mean we've measured them persist, where the numbers are basically stable in a sample that you've removed from the river. So it's not getting new inputs, and the numbers are pretty stable as you subsample it through like a month at least. So they can persist for quite a long time, and maybe considerably longer than months. And so one of the ways that connects to management, for example, is there are some places where there's, let's say, seasonal disinfection of sewage outfalls. And if you load up the sediments in the wintertime, right, then you have a source during the recreational season, potentially, if they are lasting long enough. So having long persistence is something that is, I think it's potentially a big problem. And unfortunately, it's exactly the places where, exactly the conditions that are created where you have a lot of inputs are exactly the conditions where you get more persistence. So it's sort of like, you're getting kind of double whammy, because you're getting lots of inputs, and then you also have the potential for resuspension of those inputs later on. Yeah.
Yeah, interesting. So I guess clean water would be a big help in terms of clear water, I should say, instead of clean, but more clear. And the Hudson River doesn't take much to go out there and notice. I don't think I've ever seen the bottom.
Right.
but you do see the bottom a lot in a lot of the streams. So
Yeah,
that's
for
kind of
sure.
an interesting,
Yep.
that's kind of interesting.
Yes. Yes. And I can't say that we've done a ton of work on this, but what we have done does suggest that the persistence of fecal indicator bacteria of different kinds in the streams is lower than in the Hudson, which is exactly what you just suggested, right? Where you have clearer water. And we've done experiments where we've manipulated the amount of turbidity and stuff like that. And there's definitely a very strong effect. So yeah, it would definitely help in terms of this particular water quality problem to have less turbidity. I don't know how that would happen, but it does happen naturally, right? There's a lot of embayments where the water kind of goes in and then it's like it's retained in that embayment longer than it in the main stem of the system. And that gives enough time for some of the turbidity to settle out and the water becomes clearer. And unfortunately, when that happens, the nutrient levels are so high in the water that you tend to get algal blooms. So you create a different problem. So if we somehow could, magically create a less turbid Hudson River, we'd also have to do something about the nutrients because otherwise we'd have really bad, harmful algal bloom problems. Yeah.
I wonder when they test for the beaches and they kicked up some sediment, I wonder if that would change their readings.
It would definitely change their readings. We've done that experiment. Yeah, it's
You
very
have, okay.
it's very easy to kick up the sediments. Yeah. I mean, we basically, just to simulate what would happen if kids were jumping around in the water, we put on some wading boots and jumped around in the water a little bit like little kids, measuring before and after, in a place where we knew that there was pretty high levels in the sediments. And yeah, the levels in the water went up quite a bit. Yeah, so you can easily resuspend contaminated sediments just walking through the water. When we do sampling, and what we've always told the volunteers is that you walk into the water and then you reach as far away as you possibly can, or use like a pole or something to collect water where there hasn't been resuspension, because we know that that's an issue.
Right. But is there any correlation if the Intero is avian well, if it is avian, right? Is there any less human health risk if it's avian? So is it worth
Maybe.
finding out the difference?
Oh, it's definitely worth finding out the difference because it's a very different management problem, right? I mean, there's a lot of places along the waterfront where, for example, there's tons of seagulls because people feed the seagulls. And if people stopped feeding the seagulls, there wouldn't be so many birds there, and probably the water quality would be a lot better, right? And so that's really different than, let's say, fixing the sewer pipe, right? So you need to know what the source is in order to know what the appropriate management solution is. But in terms of the health risk, there's no reason to expect that coming in contact with fecal matter from animals is any safer than coming in contact with fecal matter from humans. There are so many diseases that you can think of that are transmitted via animals, right? I mean, COVID-19 originated in bats and then was transferred to another animal host and then eventually got to humans. Bird flu, right? I mean, you name it. A lot of illnesses reach human populations through animal vectors. So there's no reason, I would say, just because it's, animal, like it's totally okay. I'm going to swim in water that's contaminated with poop just because it's bird poop. You know what I mean? Like I wouldn't do that. But it's very different management. So it's important in that sense.
Oh, great. Yeah, makes sense. Anybody else have any questions they'd like to ask Andy? I agree. Well, it certainly has been, and being involved, and I know a lot of the people on the call have been involved with your citizen science sampling, and we really, really appreciate. I think Rockland County is certainly much better off having Lamont Doherty in our county, and it is a pleasure working with you guys. It's been a pleasure working with you and Riverkeeper. So hopefully we can if there's anything we can do to strengthen or support the relationships there, feel free, let us know. Because we certainly can get one of the main duties of this committee is getting information out. So anything that's coming down the pike, keep us posted. And yeah, Margie's been very good. We've got good connections with you and Columbia University and Lamont. So it's been great. You had to start somewhere, and now that we've got some data after the years of doing this, it's interesting. And I think it was interesting too with what you said. I think a lot of people got involved with these water quality issues, hoping that they could plug a pipe up and all the trouble would go away. And that just is not the case. So I guess in one sense, it's good because maybe we have through the years taken care of the low hanging fruit, so to speak, and that those main big pipes have been clogged, and now we're down to the hard nitty gritty. And so that part's kind of interesting, and maybe we should look at this a little differently in that we have done some good work, and people, our predecessors, have done some great things back in the 60s and 70s. And to ask one final question, Andy, I mean, the one that you started with, is our water getting better?
Okay. So it depends on your time frame. You're absolutely right. I certainly wouldn't want to create the impression that we still have the same water quality that we had in the 1970s. The Clean Water Act had a really big impact. There's no question. In the places where we have data, like New York City and Albany, the contamination levels are way lower than they used to be and way less frequent than they used to be. And that's really good news. I mean, it really is a success story. It's an environmental success story. And it has really changed. I think it has really changed people's attitudes towards the river. I think I don't think anybody would be shocked if I described the past relationship with the river was basically it was an industrial sewer. And I don't think people think about it that way any longer because the water quality has gotten much better. But the problem is that we're in this kind of like middle number scenario situation right now, where once upon a time the river was so frequently contaminated that you didn't actually have to do any testing because you always knew that it was bad. Maybe sometime in the future it'll always be good, and then we also won't need to do any testing. But now we're in a situation where most of the time it's fine, actually, and just sometimes it's not. And that's where you actually really need information. And so that's, what we have tried to provide. And people seem to appreciate it. So that's, I'm really gratified to have been part of this project.
Yeah,
And
thanks.
I thank you
Thanks
guys for
very much.
inviting me as well. It's a pleasure to have spoken to you today. Thank you for the opportunity.
Yeah, great. Well, great topic. With that, does anybody have any other questions? Any questions going once? So, Andy, thank you very much. Thanks again. I've got a couple updates. And Rihanna Polanco was not able to make it.